Our next speaker is Dr. Alice Weaver. She's the program leader at Livestock Innovation and Welfare at SARDI. She grew up on a mixed farm in the mid-north of South Australia and has turned that passion for ag into a career in research. And she's going to be talking about reducing enteric methane emissions from grazing sheep. So come on up. Thank you very much.
Green button to change slides.
Yep, and that is just a point.
Okay. So you don't need to. Great. Okay. Great, thanks Annabelle, and really great to be here presenting to you all about some of the work we've been doing in methane mitigation space, particularly in grazing sheep. As Annabelle mentioned, I grew up on a mixed farm in Kapunda. I did my PhD in pig reproduction in something completely different, and I've since come back to the sheep industry, which is fantastic. So I guess we've heard a lot of background already, but what is methane? It's a really potent gas. It traps heat a lot more effectively than carbon dioxide. It does have a shorter half-life, where it's then transferred into carbon dioxide, but it has a significantly higher warming potential than CO2 over 20 years. Methane is produced by both natural and human sources. In terms of natural sources, it's released from wetlands, oceanic processes, thawing of permafrost, but also human activities have increased the release of methane into the atmosphere. A lot has come from agriculture, as we've all been hearing about, fossil fuel production, and also decomposition in landfill. Actually, I think Sarah presented something very similar to this, but it just shows this is South Australia's emissions. You can see there that agriculture is the second highest emitter of methane or CO2 equivalent. Again, you've probably seen very similar slides today, but it's just highlighting that global greenhouse gas emissions by gas is its highest in carbon dioxide. But in terms of our livestock or our ruminant species, methane is far and away the predominant gas or greenhouse gas that is produced. And methane is produced in the rumen of the ruminant due to enteric fermentation. So this slide just shows the million tonnes of CO2 equivalents produced globally. You can see a big portion of that is coming from Latin America and the Caribbean, particularly in the beef industry, which is presented in orange. And Oceania, right down the bottom, also you can see is contributing quite significantly to emissions. So this is actually a results from a carbon baseline that was done at Turretfield Research Centre, which is where I'm based. It's on the edge of the Barossa Valley and the mid-north of SA. The property runs about 4,500 sheep over about 1,600 hectares. And you can see here quite clearly, so this is a research centre. It is run a little bit differently to a commercial grazing system. But it's quite clear, I mean, it's obvious there that the majority of our emissions are coming from methane, so around 80% is directly related to the sheep that are grazing. It's, I think, important to note or to know that there's differences between methane that's produced but also intensity of the methane. And a lot of people refer to methane produced and a lot will refer to the intensity of the methane that's produced. But basically, total greenhouse gas emissions are measured in kilograms or tonnes of carbon dioxide equivalents. And CO2 equivalent accounts for the combination of gases, so carbon dioxide, methane and nitrous oxide. Whereas emissions intensity it factors in the product that's produced. So it's estimated as total greenhouse gas in kilograms of CO2 equivalent divided by the unit of product that's being produced, and that can be wool, meat or milk. So for example, for livestock, the units of product can be measured in kilograms of live weight or wool produced on the farm, and it's put there as kilograms of CO2 equivalent per kilogram of live weight. So why should we reduce methane? Again, we've been hearing about this all morning. It's important for us to be addressing this as we want to maintain our market access, not only in Australia but globally as well. There's a lot more pressure being put on our industries to meet these market demands. Farmers need to maintain their social licence to operate their enterprises. We know that methane is contributing to climate change and global warming, so there is pressure on us to be doing something about that. And this will also help to ensure the long-term sustainability of our farms. There's pressure from consumers. They apply pressure in lots of different ways. You know, they're interested in things like animal welfare as well, but carbon is becoming, and emissions are becoming a lot more important. But also a side aspect to this is maybe we can improve our production and achieve production gains by addressing methane and reducing emissions. So I'll just talk about sheep here specifically, but there's actually 2 carbon cycles in sheep, which is interesting. So on the, where is it? So on the right-hand side, so you can see here we've got grass, which contains carbon. The animal eats the grass. Methane is created in the stomach when they digest or ferment the grass. That methane is then belched out or burped out from the sheep. A lot of people think it's farted out. It's not. Most of it is actually burped out. After about 12 years, the methane is broken down into CO2, which is then absorbed back into the pasture through photosynthesis. The other cycle is focused on wool. So sheep eat the grass. That carbon is converted into wool as part of the keratin fibres in the wool. So carbon is actually stored in the wool fibre. The wool is then made into clothes. So the carbon is captured in the garments, and either it can be stored in the garments for a really long period of time, or if it's buried, it can return to the atmosphere or back to the soil. So there's actually 2 carbon cycles in the sheep, particularly merino. Methane production varies really significantly in different systems. Animals are produced in all sorts of different ways. We've got extensive grazing systems, feedlot type systems. We might have a combination of both, so grazing, animals coming into containment, which is happening a lot more now, particularly in these drought conditions. And also the methane produced by those different systems can also vary significantly. So why is that? Higher fibre diets have lower digestibility, and they require a lot more fermentation in the rumen than, other way around. Higher fibre, lower digestibility, require more fermentation to break down. Animals on pasture have slower growth rates. They have increased age to market, and they produce therefore more methane over time. And feed characteristics have a big impact on the amount of methane that's produced in the systems. So in a feedlot type system, diets are really high in energy, low in fibre, and they have increased digestibility. So the amount of methane produced from those high input systems is lower overall than what's produced in a grass-based system. And this is important to know because a lot more of our animals are produced in grass-based systems compared to feedlot. So we really need to have a way to tackle emissions in those pasture grazing systems. So if we want to reduce methane, we need to be able to measure it. It's really difficult to do out on farm, and that's where I suppose a lot of the research comes in. This is a green feed machine. It's working. Okay. This is a green feed machine. You might have heard of it but not seen one. Maybe you have seen one. These units are made in America. They're quite expensive. They're about $150,000 to $180,000. And how it works is the sheep, they come into this raceway here. This is the green feed unit. They put their head in this little box here. It reads their electronic ear tag, and then it drops a portion of feed for the animals to eat while their head is in the little box here. They need to have their head in there for at least 2 minutes to capture the gas that the animal or the sheep exhales. And then that information feeds up into the cloud, where it goes through a series of calculations and algorithms to calculate the amount of methane and CO2 that's produced. These systems are completely portable, solar powered, so we just move them through the paddocks. These are a couple of our trials that we've been running. This photo here is quite recent, so you can see the grass, the pasture's looking pretty good. Whereas over here on the right, this study was done 2 years ago, and you can see the lack of rain. We had a really awful, there's basically no grass there for the animals. But you can see the sheep putting their head into the box to access the feed. Some of the challenges with this system, it takes a lot of effort to train the sheep to use these units. They really don't want to put their head in a tiny little box. So it's really about sort of a month's work to train sheep to try and access this. It also, we can't capture methane from the whole flock. We'll probably get maybe 40% of animals if we're lucky. So it's quite challenging to use these units out in the field, but it's probably the gold standard in terms of methane measuring in the field. So another technique that's used, I haven't used this one specifically, but it's called the SF6 tracer technique. This is able to be used out in grazing animals, particularly cows. What happens is there's a little halter that sits over the cow's head. You put a little tracer, it's like a little bolus, into the rumen of the animal or the reticulum, and it slowly releases CF6 gas over a known period of time. On the halter is a little tube that comes down to the animal's nose. So as the animal breathes and ruminates, it takes little samples of gas, and then it calculates the amount of SF6 tracer relative to the methane that's produced. So it releases a known amount of SF6 gas into the rumen. That's then released, and then we can work out how much methane is being produced. So it's quite intensive and not really something that can be done out on commercial farms. This system here is called a respiration chamber. And they've developed this for cattle, sheep and goats. But here in sheep, it's basically a little box you put the animals in. They can stay in there for, say, 3 to 5 days. It's a closed system. You feed them in there, they defecate in there, and then you can take samples of gas from that box, every 30 minutes if you want to. Again, really labour intensive, not really suited to grazing systems at all. You're taking the animals off the pasture to put them in this box. And some of you may have seen these systems already here on the top right. These are called portable accumulation chambers. So these are similar to a respiration chamber, but the animals are only in there for a short period of time, so maybe up to 40 minutes. These are portable, so there's a project at the moment being run by UNE where the chambers are going out onto properties. They're putting sheep in the chambers for 40 minutes and they're measuring methane across a large number of animals to help feed into developing a breeding value for methane. Again, there's limitations with this because you have to take the sheep off the feed for an hour before you put them in a box. And also it's only taking a snapshot of methane production over a day. It's not taking into account methane production over 24 hours. We can also look at modelling methane production in the lab or in vivo. So this system here, this is called an ANCOM gas system. So we've set one of these up in our lab at Turretfield. Basically, we've got these little bottles here that have got a gas measurement system on the top. We put buffer and supplements into this gas production module. Then we go and collect some rumen fluid. We go and get it from the abattoir. We can use sheep rumen fluid or cattle rumen fluid. You can also use rumen fluid captured directly from the live animal, either by taking a sample by stomach pumping, or you can use cannulated animals that have a little bung straight into the rumen. The beauty of this system is we can screen lots of different additives or pasture types and varieties to see how well they reduce methane or what their methane reduction potential is. So that rumen fluid then is strained. It actually goes through a food processor to mush it all up, and then it's pretty gross, gets strained and added into this module. That's then incubated at about 40 degrees for 24 hours. And over that time, the gas is collected from these modules on top, and then we can take it off and measure methane. We also measure gas production from these little capsules. Okay, so this is the ANCOM system in action. So you'll notice there's a syringe on top that's got a little plastic tube connected to the module. And when it builds up enough pressure in the little closed system, it actually produces the gas and it fills up the syringe. So we can then take that syringe and analyse it for methane. So again, it's just one of those tools we have to measure methane. It just gives an indication of how different feed additives or pasture varieties might influence methane production. So how can we reduce emissions in grazing sheep is one of the big questions that our research group is trying to ask. So our objective is that the sheep industry in Australia, it really requires a range of different methane mitigation approaches and strategies to significantly reduce methane emissions, raise the productivity of animals, and also be safe to animals, humans, and the environment. The strategies that are developed to be used on farm, they need to be scalable and economical. They have to be easy to implement in a grazing system, particularly Australian grazing systems. And I think the key here is that they also increase productivity as well. That means that it's going to be more attractive for a farmer to implement them on their property, and it reduces the risk. So how can we reduce methane? There's lots of different ways that we can address this, but probably the biggest one and the easiest one and the most attractive one is to increase the productivity of our animals. Sarah spoke about this. I think Penny will probably touch on this as well. But we can improve or reduce emissions intensity by breeding more productive animals and having more productive animals across our system. So how do we do that? It's probably what you're all trying to do already, retaining those high-performing animals, selling those animals or culling the animals that are not performing, that are not getting pregnant or are losing lambs, not raising a lamb to weaning. We can also work on enhancing the fertility and the fecundity of our animals. So can we select for animals with an increased twinning rate? Pregnancy scanning is quite important here. The use of EID technologies to track those animals. Can we breed our animals earlier? An animal is a young animal to breeding is technically non-productive until she produces a lamb or a calf. So can we make those animals a lot sooner and get them producing quicker? Also, improving animal health and welfare. A healthy animal will grow faster and be more productive and therefore produce less methane emissions in their lifetime. And I think this is where things like EID technology and herd management software, flock management software will become really important in tracking these animals. So what are some of the other options out there apart from improving efficiency, which a lot of producers are already doing? It's been Richard Eckart spoke, not Richard, Richard Heath? Richard Heath, sorry. Richard Eckart, the godfather of methane. Genetics, there is a lot of work starting to be done now on developing an EVB for methane production. So can we actually select and breed from those animals that produce lower methane? There's also a lot of work being done into pastures that reduce ruminant emissions. Different pastures have different bioactive compounds. Some of these compounds you've probably heard of, which are tannins and saponins, and they can actually help to reduce methane from ruminants. There's a few species that SARDI is looking at particularly as part of a CRC project, which is lucerne, vetch and medics. There's also a fair bit of work looking into manipulating the rumen, and there's different ways that we can do this. So researchers have been looking at a bolus that's implanted directly into the rumen. It slowly releases antimethanogenic compounds over time. And I guess the beauty of this is you can administer it to the animal once, and then over, say, 6 weeks, it will reduce methane. So it's a one-off treatment to reduce methane. Some people are looking at vaccines to reduce methane. And then there's things that we can put in the animal feed. So feed additives that reduce methane, they're known as antimethanogenic additives. And we can also look at things like pre and probiotics, which are getting a lot more interest. So I'll just run through some of the projects that we've been doing at Torretfield, looking specifically at reducing methane using antimethanogenic feed additives. This is done under the Merrill body of work, which is methane emission reduction in livestock, which is a federally funded program of work. It's a collaborative project as part of the National Sheep Methane Program. So we work with collaborators at the University of Western Australia, the University of New England and New South Wales DPI. And the work is also funded by, so AWE contributes significantly financially, but we're also working with commercial companies that are already producing these feed additives. The photo there is just of the project team last time they were here in South Australia. And I'll just acknowledge Elise Lowe, who's in the office, in the room here, and Jamie Daly, who have been doing a lot of work in this project. So we've been looking at a range of different feed additives most of them are commercially available. There's a lot of data already on these, but it's important to note that the additives have been studied more so in feedlot systems and mostly in cattle. There's really limited work that's been done in sheep, particularly in grazing systems. So we've looked at 3-nitrooxypropanol, otherwise known as Bovair. This is a synthetic product that's manufactured in a lab, and it works by inhibiting the enzymes that produce the methane in the rumen. We've also looked at synthetic bromoform. So bromoform is the active compound in Asparagopsis or red seaweed. You may have heard about that. There's a lot of information on Asparagopsis. It's often in the news. But we worked with Ruminate on a synthetic formulation that they had for it. And that one was also water soluble and water stable. So we were able to deliver it through the drinking water. Agalin ruminant, this is another compound that's been around for a while. It's actually used to improve the productivity of the animals, but there is signs that it reduces methane. Agalin is a blend of different essential oils, and it works by modifying the microbes in the rumen. So rather than actually inhibiting the methanogenesis process, it actually changes the microbes in the rumen. Nitrates is something that we'll be looking at later this year. Nitrates act as a hydrogen sink, so they actually take up that hydrogen and use it so that the microbes can't then turn it into methane. I guess the thing to keep in mind with all these different feed additives is they have different modes of action, and they also have different duration of effect. So an enzyme inhibitor will have a very short effect, so you'll feed it, you might reduce methane for 3 to 4 hours, and then it will go up again. But a rumen modifier, it sort of starts or it affects methane a lot more slowly, but it's cumulative and it builds up over time, and actually it stays active for a longer period of time. Looking at these combinations either alone is good, but you could also look at them in combination, and that's something that a lot of trials are starting to do now, particularly ones that have different modes of action. So this first study, I'll just run you through. This is a pilot study. We were really interested in delivering antimethanogenic additives through the drinking water. We used 120 dry ewes over about 120 days. It was run over summer while it was quite hot. And we wanted to focus on water-based delivery. It hasn't really been done much before. A lot of work has focused on antimethanogenics put into total mixed rations or feeds, pellets to animals in feedlots. We looked at 2 methane mitigation additives, Agalin ruminant and the bromoform, which was water stabilised and soluble. And so this is the layout of the trial. We had 6 paddocks used in this project. So the 3 treatments were replicated twice. You can see the lines of dividing the paddocks are in red. In the blue is the water troughs for each paddock. So they all ran off a single water line. This down here is our water system that Elise worked very heavily on developing. There are a few companies out there that will do automatic dosing of water, but we wanted to do a controlled closed system, so we had a lot of control over the amount of additive that was being delivered to the stock. And also we were looking really carefully at the drinking behaviour of the animals. This here, we have a EID reader that's fitted over the end of the water trough. So we had 20 sheep in each paddock, and as they came up to access the water trough, it just read their tag. So we got some really interesting data on water intake and drinking behaviour of the ewes. Again, it's all solar powered, so we just plugged it and played. So here's a ewe coming in to access the water trough. We also measured methane from these animals using the green feed, and these animals are pretty well used to the green feed and us by the end of the trial. So in terms of I'll just say as well that this project was a bit more of a safety trial. So we were more interested in could we provide these additives through the drinking water and not have any negative impacts on their production or their health. There's a lack of information on how much the animals will be drinking and what's a safe level to include these additives at. So that was probably the key outcome of this trial, is looking at the safety. But we found some really interesting results with water intake. So up the top there, you can see this is the drinking behaviour from ewes that received either no antipathogenic additive, agalin, or the bromoform through the drinking water. And the key thing to point out here is that, sorry, this is very awkward. The key thing here to note is that water intake did not differ between the 3 treatment groups, which was good. We didn't have any impacts on palatability. There was no difference in the water intake per ewe per day, but we did see a big difference in the number of visits to the water trough. However, important to note that they still drank the same volume of water over that trial time. So the control animals were visiting less frequently compared to the bromoform animals. They still drank the same volume of water. And we think this is probably more related to trees and shelter in the paddock. So we found that if animals had access to shade, they'd spend more time sitting in the shade. They'd visit the trough less frequently, but drink a bigger volume of water at each visit to the trough. As opposed to animals that had little shade, they visited the water trough a lot more frequently. Okay. Basically, we saw no differences in body condition or wool growth. We saw a slight reduction in wool growth in agalin-treated animals, but it wasn't too much of a concern, and it probably wasn't the primary role or primary outcome of this trial. So good outcome there. This is the really exciting result for us, is that we did see a really good reduction in methane emissions. So the treatment in the middle, the agalin, we reduced methane by about 40%, and the bromoform treatment also reduced methane to a lesser extent. I'll go over this really quickly. Yeah, how much time do I have? Okay, all right. I'll go quite quickly. Okay, so this next study was looking at the product Bovair, or 3-NOP, and we were looking at delivering this through a pellet to see if feeding frequency impacted methane emissions, and we wanted to know if feeding it once, twice, or 3 times a day affected the emissions of the animal. So we had 250 breeding ewes and their progeny. The 3-NOP was incorporated into that pellet, and we delivered it via those automatic feeders, which meant we could control when the feed was dropped and how much was dropped at each feeding bout. And it's important to know that the total volume or total amount of 3-NOP was the same. It was just split over different feeding frequencies. We looked at methane production and productivity. And this, I'll just give some quick results, but the methane, we didn't see a difference in methane when we fed it once a day, but we did see an impact or a reduction in methane when we fed it twice a day. And we think this is probably due to the fact that it has a short mode of action. Just keep in mind, this is methane over 24 hours. So because it only has a short period that will reduce methane, over the rest of the 24 hours, it probably caught up. I'll just skip over live weight and body condition score. Basically, there were very minimal effects on it. So we did see that 3 NOP could be delivered to sheep via feed. We did see that feeding frequency impacted methane production. We saw minimal impacts on health and productivity. We did see some slight variation in ewe live weight and body condition score between the groups. And the animals that were fed 3 NOP twice a day had a really slightly reduced lamb survival at birth and at 7 days, which then led into some effects on lamb birth weight. I'll just quickly mention this trial. So this next one we're doing is looking at delivery systems. So how can we optimise delivery of antimethanogenics to grazing sheep over in extensive systems? So we're going to focus on loose lick delivery and also build on the water delivery. And we will look at agalin ruminant again, Bovair and nitrates, either alone or in combination. Thank you.
Absolutely fascinating, Alice, and thank you very much. And if you would like to catch up with Dr. Alice Weaver at afternoon tea, I highly recommend. We won't have time to do questions at the moment.